thanks again, Jesse, and thanks for all the great questions. Check out the Slack for further discussion. So we're going to move on to the final talk in this session, our first keynote from Dr. Wolfram Schultz. Dr. Schultz received training at the University of Heidelberg before postdoctoral stints in the United States and Europe. He's currently a professor of neuroscience and a Wellcome Principal Research Fellow at the University of Cambridge. He's also a fellow of Churchill College and the Royal Society. So I think the work of Wolfram Schultz is probably familiar to a few of you potentially in this audience today. And so primarily the seminal discoveries related to reward prediction error signals and dopamine neurons that form the foundation of a lot of the work that many of us do. So we're really excited to have Wolfram here. And Wolfram, you have 45 minutes for your talk. And you can go ahead and share your screen and get started. Thanks.
Should be shared.
Yes, you shared. Yes.
Can you see my first blue slide?
Yes.
Perfect. Thank you very much. And thank you for this magnificent idea of doing this conference on the quick on the internet. It's very challenging. We'll see how far we get. I'll be talking a little bit about more about the background and then going to the current work that we're doing. You see on the left the collaborators over the past years that have contributed experimentally or conceptually to the work. As I said, we start out very basic with our Parkinson patient that tells us that the dopamine system is related to movement and maybe to cognition. What you see is the eye position of a monkey when it views a stationary environment for 14 seconds. On the left, the control. In the center, when 95% of its dopamine neurons are destroyed by MPTP. And on the right-hand side, when the same monkey receives L-DOPA. So visibly, what you get when you measure the movement is the movement deficits. All right, this is the standard dopamine picture. And the question is whether these prediction error responses would suggest the role of dopamine, of these dopamine processes in learning or in decision making, and or learning and updating decision variables, and whether that may also, the dopamine neuron may be also involved in generating approach behavior and choice. The discussion is open at the moment, and there are strong arguments in both directions or against both directions. One of the, oops, I'm trying to get to the next slide. Here we go. Yes. The recent experiments we've done were on infecting dopamine neurons with channel rhodopsin and stimulating them. And we had a stimulus, the blue one, associated with optogenetic dopamine stimulation on channel rhodopsin, and the red stimulus not associated with anything. And what we see that over repeated trials, the animal comes to prefer the blue option. That's the one that's stimulated with the optogenetic stimulation. The electrical stimulation has also an effect, even stronger. Of course, it's very difficult to compare the impact of these two effects because electrical stimulation directly opens the voltage sensitive ion channels. The interesting thing then is that if you pair, for example, a reward with optogenetic stimulation, that's the center image, or you just give the same reward without the optogenetic stimulation, whether you get an enhanced CS response to the cue, to this blue cue, which now is associated with a reward plus the optogenetic stimulation as compared to the red cue that is associated only with the reward. And the answer is yes. So the optogenetic stimulation, the excitation of dopamine neurons, is a viable mechanism for both behavioral learning, in this case on the right-hand side for the preference learning, and for the neuronal learning. That is quite nice to understand what the potential role of dopamine neurons is in learning, because there's still ongoing debate. The next slide shows that we have tried to use this transfer from the US to the CS that is that we showed before for learning probabilities. So dopamine neurons are increasingly activated by higher reward amounts. That shows the left slide, that's not surprising. But we understand that reward value is simply the product of reward amount times how often the reward occurs, probability. So we take that graphic scheme of the US to CS transfer that underlies Pavlovian learning and try to apply that to the so-called frequentist approach of learning probabilities. And so the top row with these lots of blue dots shows very frequent rewards. And then the row below is some rewards, and the bottom row is very few rewards. And that will be, the idea is then this should be transferred into probabilities on the left-hand side. And here is the behavioral experiment. The animal chose between a cue that was associated with a fixed, with a reward at one single probability, a familiar reward, familiar cue, probability of 0.5. And on the left were the novel cues, which 3 cues had different probabilities. So the animal always chose between one novel cue on the left and the familiar cue on the right-hand side. And we changed these novel cues repeatedly. And what we see is that the animals initially go always for the novel cues. They're very interested in the novel cues. Whatever the novel cue may mean, they're very curious to get that. And then after a while, their choice of the novel stimulus deviates or distinguishes or differentiates according to the different probabilities. Of course, the animal chooses more frequently, the more frequently rewarded cue. Okay, the neurons learn that, and this interesting. There is an initial response to the stimulus, to this novel stimulus now, that deteriorates with the novelty and does not distinguish between probability. So this is clearly not a reward response. That's a novelty response. And the second component of that two-component response then is what we call the standard dopamine prediction error signal that codes the value, or as now would have said, the first derivative of the value, the prediction error. And we see that this signal differentiates over trials into, according to the 3 probabilities with which the reward would occur. So now I mentioned that there are 2 response components, an early one and a later one. And I want quickly to talk about that because that has created substantial confusion in the literature. Such multi-component responses are not infrequent in other systems. Dopamine is nothing special. When you look through all these graphs that I show on the left-hand side, cortical and subcortical neural responses, there is often an initial response that's non-differential and a later response. Then, for example, the target distractor on the left-hand side, after a while that differentiates. And one sees the same thing in the reward system with rewards on the right-hand side. So the 2-component response is something that happens in the brain. In the dopamine neurons, we found out at least 4 parameters that modulate that response. One is the physical intensity. A 90 decibel noise produces a stronger dopamine response than a 72 decibel sound. A reward context, when a small picture, a big picture, or reward occur in random alternation with the same background in the same context, the neurons respond to all 3 quite substantially. When we show distinct contexts in blocks of trials with different backgrounds, we see that these responses distinguish or differentiate quite well. And what remains is a substantial reward response, and maybe this some response to the big picture. Similar with randomly alternating aversive and rewarding stimuli. So here's a wonderful response in actually 75% of neuron to a conditioned aversive stimulus, a little light emitting diode. And the reward stimulus is also a visual diode with a little different color, okay, green versus red. When then we change the reward stimulus, but we keep the aversive stimulus identical, then the aversive response deteriorates very much to about 15% of neurons remaining that are activated by this aversive stimulus. And then the novelty response that I just showed, when the animal shows eye movements, shown on the top with these traces, in the first trials with a novel stimulus, the neurons respond. And then a few tries later, when the animal doesn't care anymore for the stimulus, the neurons don't care for that stimulus anymore either. So what I'm trying to say is there are situations in which dopamine neurons will respond to non-rewarding stimuli, but they're in one way or another related to somewhat reward. We would think this is a salience response, but the salience response is not all that what dopamine neurons do. It's just the first component, and we think this is very closely related to some reward in the environment. So a very controlled environment will be needed in order to separate this out. The second component then is our standard reward prediction error that we're usually showing. Okay? So the question is, what is dopamine doing? I mean, this is a question that you hear absolutely everywhere. And I'm just very, very stunned by that question. And we can give three answers. What I just showed is this fast prediction error response with an initial salience component, what I call the stimulus detection here. And we'll talk about that word utility a little later. The second is the slower response. Anything that is slower than this initial phasic response, including now Uchida's ramping response that he showed. And then, of course, the tonic presence of dopamine that's necessary to prevent Parkinson's disease. Okay. So I like to talk about this slower component a little bit, because again, that is sometimes difficult to grasp. So here is one of our very early responses, a couple of hundred centuries ago, in a, I would call that for a monkey, a fairly complicated task with a lot of stimuli going on and the go, no-go situation, lights and everything. You see this monkey there. You now understand why I couldn't become an artist and had to go into science. That I can't make a career on that graphic capacity. On the right-hand side, we see quite differential, I wouldn't say that, I would say diverse responses of dopamine neurons that go all over the place. This looks like a striatum neuron or a frontal cortex neuron. This is what I call diverse in dopamine neurons. The reward prediction error response is fairly similar between neurons. You can call it homogeneous or not, it doesn't matter. It doesn't concern all neurons, but it concerns anywhere between 55, 60% to 95% of neurons, depending whether you are in the A8 or you go over to A9 and A10. The slower response, slower and lower response of dopamine neurons is very diverse, and you see it concerns subfractions of dopamine neurons. You can have an excitation, you can have an inhibition, you can have that around the movement, you can have that at the times of reward, and you even get a certain percentage, but if you keep counting, you'll find them, neurons that are tonically activated between the trial start and the reward. It's not the nice ramp that Now had shown, but it's still some tonic activity, and you can see from the rasters, any statistics would become significant here. So this is a different thing than the phasic responses. And we thought we had found something for Parkinson's disease, a correlate for Parkinson's. Fortunately, we couldn't get that paper published. And oops, I'm still having difficulties here. Yes, but we looked at the details. We looked at the muscle activity during that task in the monkey. And you see on the left side, plenty of muscles are active around the time of the movement, indicated by key release. In the center top, you see even the contralateral muscles are to some extent excited. And these are rectified EMGs. And to the bottom, you see that the mouth movements, the licking is also quite active when the animal gets this reward, but also before at the instruction. This is several seconds away from the reward. So this animal is doing a lot of stuff here. And even the eyes are going all over the place. And of course, when the eyes move, you have a movement in the visual scene, you're producing visual responses on the retina. So we had a lot of stuff going on, right? And then what we found out later is Mail and DeLong came, I'm talking of great old history, and couldn't find any activation with dopamine neurons. And we also had problems to replicate our data when we scaled down the experiment. You see at the bottom an arm movement that occurs, in fact, to the offset of a stimulus, which is still a movement, but a sluggish movement, and you see absolutely no activity. You had 16 neurons, none of them did anything useful. And with eye movements, we never found anything. Okay, so we had a problem. We had an irreproducible result where we thought we had found some movement relationship of dopamine neurons. Unfortunately, optogenetics came along. People were working on rats and mice and found the same slow activation that we have described. We fortunately have coined it not specific movements, but we have called it behavioral activation. So look at this rodent rat on the left side in the T-maze. This animal has gone around down that alley and then turned to the right, hopefully, with hundreds of muscles active. Or the treadmill running on the right-hand side. This animal is just activating 90% of its muscles while it's running around. So this is a massive activation that involves muscles, that involves sensory stimuli. Then of course that involves acceleration. If the animal accelerates, the behavioral activation will increase. Just like when I start waving my hands all over the place, I have a higher behavioral activation. And you see on the right-hand side, even the movement start on this thing, even if it's less than a second duration, will produce, in this case, the depression of dopamine activity. So I think, and this was a bit of an early conclusion that I made in that slide here, I think that we are dealing with, schematically spoken, with two processes in dopamine neurons. And one of them is this reward prediction error. This is the phasic response, very short, much shorter than anything else, less than 200 millisecond duration, 150 maybe, and but quite high. Instantaneous frequencies can go up to 20, 30 impulses per second. And then the slower activation, some of which have been talked about much more specifically by now an hour ago, okay? So I think we have maybe two processes in dopamine neurons that we want to keep apart. We can talk about the reward function and the motivation function. We can talk about the reward function and the behavioral activation function, which is what I prefer. It doesn't really matter. But what we want to understand is that we have quite substantial heterogeneity in these dopamine neurons. So what were we actually doing? We were asking, what is dopamine doing? That was the naive question. Now you read this beautiful text by Douglas Adams, who is another Cambridge guy. And he found out, he figured out, he devised a computer called Deep Thought that calculated the super question to the universe of the life, universe, and everything. And after a few million years, the computer announced 42 was the answer. So we got some answers, but then another bigger computer had to be built to find out what the question was. So our question was, what is dopamine doing? And I'm not sure that's a great question, because that question is based on what I now think is a misunderstanding, that one brain system means one function. I just think that this may be true for the retina or for the motor cortex, but it may not be true for the dopamine neurons. I don't think we should maintain that idea, and I don't think that I like to keep asking that question, what is dopamine doing? Okay, dopamine does several things, and if you can try to bring it together in one coherent idea, the better. Okay, so another thing that causes issues is, as Now has mentioned, the temporal difference learning model, in which the reward response, the prediction error response, does not only occur to the reward, but also to the conditioned stimulus, but even to an earlier conditioned stimulus. All of these three red responses are prediction error responses relative to the prediction that was active at that time, right? And when for example, the CS1 stimulus occurs and then the reward occurs, there's no more response at the time of the reward because that reward is predicted. When the CS2 occurs, and then the CS1 occurs, then there's no more response to the CS1 because the CS2 has already predicted that CS1 response, understanding that CS1 and CS2 are now higher order rewards, okay? And that of course comes from the idea that an animal would follow the trace of behavior that brings in the largest reward. So in that graph at the bottom, the animal would go from the initial prediction V at t-1 to the largest reward, and then to the next largest reward, and next step to the next largest reward, okay? So if we understand that actually the scheme is a bit more complicated than the simple Pavlovian CSUS situation, and then we go into more complex real-time, real-life tasks, we figure out that we can find dopamine neurons in every single of those studies that I'm showing here. Some studies say it explicitly, in particular our studies, because we started out these studies from that idea. But then there are other studies where you find it when you go through the figures, even though the studies may keep a question on it or may not even mention it explicitly. Dopamine neurons seem to code these TD errors in so many situations that it's very difficult to identify them. And then it becomes very confusing and you come up with wonderful cognitive interpretations of dopamine response functions. So here's an example where Minoru Kimura has done a very substantial and time-consuming analysis of dopamine responses in his sequential task, in which 3 events with different reward probabilities succeeded each other, and the sequence could stop at any of these 3 steps. You see on the right-hand side, the dark line, the black line, that is his TD model. Of course, he adapted, optimized the parameters and learning rate and maybe also the gamma. And the bars are the dopamine responses. And it looks very nice that it fits quite well that model. And there was just this recent paper, I think it was last year, by Ben Engelhardt, who's looking right at me now, in Nature, where they had a very substantial multiple linear regression in order to come basically to the same thing with a few other additions to that, where in a complex task in a mouse, they could identify these TD prediction error responses through that analysis. So just to wrap this up, the overview, the whole thing of TD prediction error and dopamine started initially with, unknowingly, with a Bellman equation that defines the optimal value function, in which the value function is defined by the maximum of reward that you're getting from the current reward plus the discounted sum of all future rewards. And then the TD model is simply that Bellman equation minus the green VT minus the current prediction. That's the TD learning. And that, of course, is an extended Rescorla-Wagner learning rule. And you see the similarity, the way I wrote it, in the equations. And that, of course, goes back to this ridiculous experiment by Pavlov, in which the dog salivates to the bell that rings, has rung repeatedly while a sausage had been presented to the dog. And then at the end of the day, even without the sausage, the dog salivates to the bell. So Pavlov 100 years ago is actually at the beginning of this temporal difference learning, which then resulted with Demis Hassabis' work, which of course added many other machine learning tools to the TD prediction error learning rule in a fantastic Go game, performance better than the best human. On the right-hand side, you see the blue curve, and that was in 2017. And from what I understand from Demis Hassabis is that the performance has increased even further, and it has also been applied to the chess play. Okay, so we're seeing this step from Pavlov to this very nice artificial intelligence Go game, of course, with the addition of other machine learning tools, just like the dopamine neurons are not alone in the world, and they have the whole striatum with its very complex structure around them, supporting their function. Okay, right. Well, that's interesting, but for me, from the biological point of view, not from the formal modeling, but from the biological point of view, I have a little issue. And the issue is that biological organisms are not silicon machines. Reward value is subjective. Of course, it's programmed into the brain, but it's still a different thing. On the left side, we see a somatosensory receptor that is sensitive to deflection. So these sensory receptors translate the impact of external physical signals like intensity, velocity, acceleration, whatever you want, photon impact into neuronal signals. Right? For reward, there are no receptors. There are no receptors because we eat a steak now in the US. You're going towards lunch now. You may have your steak for lunch. You may have a second steak. I'm sure at the third stake, you're going to stop. However, physically, the stakes are exactly the same. I actually copy pasted them here. And the stakes produce exactly the same stimulation of the mechanoreceptors and the taste receptor, whatever you want. Still, they're not a reward. So the reward value, rewards don't exist. There's nothing, no such thing in the outside world that is a reward. There's no receptor for a reward. This is all made up by the brain. And of course, the brain works with signals. The reward signals are made up by the brain as well. So it's interesting. The brain generates signals for something it makes up itself. Anyway, with that, we have a start to understand what these economist fellows did a few hundred years ago. We need a description of subjective reward value. And I don't like these ad hoc definitions of a little temporal discounting here and a little satiety here. What I want, what I'm looking for, and it's right here. It's 280 years old, a mathematical function that has predictive value. Once you have a function for reward value, for subjective reward value, you can predict choice. That's the idea. The value-based choice is predicted by these old economist functions. The Bernoulli function is the scalar utility function. The Neumann-Morgenstern utility is utility under risk. And we're going to use this utility under risk in order to establish a utility function. Before we do that, we need to make sure our animals understand what we're doing, because we're going now into subjectivity. We need to believe what the animal is telling us. I hope they don't lie. And we have no way of telling us unless we have good behavior. What I'm using now is what's called first order stochastic dominance, a very fancy thing for a very simple thing. And it says simply, if you have multiple options to choose from, all options are the same, but one option must have at least one better outcome, or no worse outcome, at least one better outcome. That would be the stochastically dominating option. Stochastic by meaning there's some uncertainty involved. We'll see that in a second. So here's my example. You have a person choose between $1 for sure, or $1 or $2 in every 50% of trials. So the person would get at least $1 on the right-hand side if you choose the right option, but it might get in 50% of cases a second dollar in addition. Very simple, right? Everybody knows this is rubbish. You will never choose on the left-hand side. And that's completely wrong. I'm very sorry. I did that test with one of my neighbors, who is the wife of a banker, retired, and she failed the test. Because she said, we never do anything that's risky, because the right situation is risky. Note that you will always get at least $1, but it's risky in terms of you may only get $1, you may not get the $2, right? And of course, if you listen to the discussion of face masks at the moment, where people say, well, the face mask may not work, so I won't use it, that is clearly a violation of first order stochastic dominance, because the face mask doesn't cost anything. And even if it works only in 10% of trials, it's at least 10% of trials better than no face mask. Okay, right. So this is what is, this is a violation of rational choice. The decision maker gets a suboptimal outcome. What do monkeys do? Right? Here we give the monkey the same choice. On the left, the blue rectangle, there is a little bar. The higher that bar in this rectangle, the more reward the animal gets, fixed reward, right? So the blue one, the blue option shows a small reward in every single trial when the animal chooses this, in this case with this eye movement, onto that blue rectangle. The red rectangle gives either the same reward in 50% of trials, and in the other 50% of trials, it gives the higher reward, a substantially higher reward, right? The animal will always get one reward when it chooses the right option, but it may be the larger or the smaller. It will never get both at the same time, okay? But clearly, the right-hand option is first order stochastic dominating the left option. Very clearly, something is better in the red option. The monkeys understand that. They are not 100% sure. They do repeated choices. They do a few dominated choices. That's the question of stochastic choice, which is something different than the stochastic stimulus, okay? But of course, this may be explained by the animal being risk-seeking. I showed you that the animals are novelty-seeking. So it could be that the animals are simply risk-seeking. That's why they choose the red option. All right, let's do the control. The blue option now on the right-hand side has the same risk as the red option before, but now the left option here, the safe, high red option, is of course better than the blue option. If the animal were risk-seeking simply, it would choose the right option. If the animal is obeying first-order stochastic dominance, it will choose the red option. And you see that in 75, 80% of trials, the animal chooses the better option. The animal is observing first-order stochastic dominance. And dopamine neurons do exactly the same thing. Now, the two stimuli that we present, either the red one or the blue one, are slightly different than the left side because we wanted to have symmetric stimuli, right? But you see on the right, on the stimuli on the right-hand side, with the dopamine neurons, that the blue option has the lower outcome is a little higher than the lower blue outcome, right? So the red option is better because the top outcomes are identical, and the lower outcome is better for the red option. And the dopamine neurons respond stronger to the red option than to the blue option. And we see the same thing in choices. Okay, good. All right, that is the first thing. It looks like our animals understand what we're trying to do, and the neurons are understanding what we're trying to do. That means they understand the stimuli, and they're actually even rational. Of course, they have done 20, 30,000 trials, no question that they are rational compared to my neighbor whom I'm meeting at the bus stop and asking one single question, okay? So let's see if the neurons or if the behavior is actually following the subjective value called utility, or they're following physical value. So what we do is we give the animal a choice now between a safe stimulus on the left and a gamble that where either the top or the bottom outcome will occur with 50%, okay? And then we do a psychophysical titration in which we keep the gamble constant and we move the safe outcome up and down. All right, so here now the safe outcome is quite low. You see the gamble at the bottom between 0.1 and 0.4 milliliters. Okay, that's the gamble. It's a binary gamble, 50% each outcome. The mean, the so-called expected value, is right in the middle. Right, that's the mean value of the gamble. We are moving the left stimulus around in the psychophysical function. It's a very short function I'm showing here, a test. And when we move the left stimulus a little higher, the animal chooses the left stimulus a bit more. You see at the left, the probability of choosing the safe reward. That's this single bar blue stimulus. And we go down, it chooses less. We go higher, it chooses more. And we draw a function through it. We fit a function through that. That looks like an ideal fit, a little optimized. And we see that this function crosses the probability of 0.5 at one point here. And at that point, the animal chooses the safe outcome and the gamble with equal probability. That means for the animal, the gamble and the safe outcome have the same value, right? Same choice preference, same value. That means that actually the safe outcome is well above the mean. We see the CE, the so-called certainty equivalent, is above the mean, is above the expected value. That means the animal puts more value into the gamble than into the safe outcome. The animal is risk-seeking, okay? At that point, the animal chooses these, as I said, these both outcomes at the same frequency. But if you realize, if the animal chooses the gamble 50%, it's actually getting less reward compared to the safe outcome. So the animal is foregoing some physical reward in order to do that choice. And there's only one explanation, is that the animal is choosing not according to the physical value, but according to the subjective value, which we call preliminary utility, okay? Well, Daniel Bernoulli said 280 years ago that value is not an explanation for choice, it's utility, this concave utility function that I showed you before, okay? What he didn't know is that dopamine neurons are doing that job. So on the left-hand side, I plotted 3 gambles, three identical gambles at three different levels. And we see that the green gamble now, that the subjective value is exactly where the expected value is, but the orange gamble has a lower subjective value than the mean. That means at the orange gamble, at the higher range, the animal is actually risk avoiding. And with the green gamble, the animal is risk neutral. So we have a chance, a change from risk seeking to risk neutral to risk avoiding. And we'll explain that in a minute. Okay. And you see on the right-hand side that these responses are also identical, just like the animal's indifference point. The dotted lines are the safe reward, and the continuous line are the gambles. And you see that these neurons are, of course, sensitive to the amount of reward, and they're increasing this from blue to green to red. Okay. Right. So what we really need to do is we need to get a utility function now, a real function. We do the same kind of choices, titrate them. We use what's called a fractal procedure. It's a repeated so-called chaining procedure in which we take a gamble that is 0 and 0.1, 0.2 reward as a gamble, and try to get the certainty equivalent, the indifference point, which is this blue arrow, which is at 0.75 or something like that. Then we take that point as the top gamble point and get a new certainty equivalent. The certainty equivalent, of course, has 50%, has half the utility of the out of the top and bottom gamble values. Okay, and then we continue doing this, and then get this very funny utility function, which is initially convex, then becomes linear, and with higher values becomes concave. And then we want to know what dopamine neurons are doing in that situation. And here we go. The red curve is the utility function in that same monkey in which we record neurons, the dopamine neurons. The dotted red line is the expected value, the physical value, and the gray bars are the dopamine responses to unpredicted reward, right? This is the most basic, simple, stupid reward prediction error. You produce a reward out of nowhere and the neurons respond always because it's not predicted in time, even if it's the same amount, right? So what we see is that the black bars follow much better the red curve, not ideally, that's biological noise, but much better the red curve than the red dotted line. So we are seeing some utility signal. Okay, all right. That's okay. That's an interesting result, but it's not what we really want to know. What we want to know is our reward prediction error signal. So we understand if you take a gamble and the animal gets we take the left stimulus as the predictive stimulus, show the animal only that gamble. If the animal gets the top outcome, it will be a positive prediction error relative to the predicted mean, the expected value, right? And we would get an excitation. If the animal gets the bottom outcome, it would be a negative prediction error because it's less than the predicted mean, and the neurons would show a depression inactivity, okay? It gets complicated. So we're just using the positive prediction error. It's also a stronger response, much easier to quantify and a bit less noisy, okay? So what we do is we take our utility function, we plot three gambles onto that utility function. The three gambles are, as you've seen from before, for example, the red gamble is from 0.1 to 0.4. So the bottom outcome is 0.1, the top outcome is 0.4, and the range is 0.3 milliliters, and the positive and negative prediction errors would be 0.15 milliliters physically. And we plot these gambles on different parts of the utility function. And then we look at the utility. We're going from the x-axis to the y-axis. We want to have the prediction error not in physical value, not in 0.15 milliliter, but in utility, right? And then we see that because the utility function in the center with the black part is more steep, that the utility prediction error for the center gamble is higher than for the red and blue gamble. And then we want to see if the dopamine prediction error response would also show this non-monotonic change. And here it is. The black response to the larger utility prediction error is higher than the red and the blue response. And what we get then is, as a result, the dopamine reward prediction error can be specified as a utility prediction error. We want to have a metric on that reward for the reward prediction error. And the metric is now not physical amount, we've seen that, but it's utility. And then we can reformulate the what's called a Wagner learning rule and the tempered difference learning rule in terms of utility prediction error, okay? So maybe one more slide, and then maybe two, but we're running out of time now. Well, I have five more minutes. Yes. All right. So here are two
You've got
gentlemen.
5 minutes.
Yeah, thank you. Here are two gentlemen to whom we are eternally grateful. Peter Bossarts alerted us when we had created already the red stimuli on the right-hand side that these two gentlemen had produced a wonderful theory behind that. That's the most simple economic risk definition that one can find. We're talking of economic risk, which is based on the uncertainty, not on the loss risk that the insurance uses, which is the probability of loss. This is not what we're talking about. For us, the red gamble is more risky than the blue gamble because the outcomes are more uncertain. You don't know whether you get a very small or a very large reward with the red gamble, whereas with the blue gamble, you get a reward that's a little bit smaller, a little bit bigger. It's not a big deal, okay? So the red gamble is more risky than the blue gamble. And these two gentlemen had a very nice article many years ago, actually 50 years, where they showed this has the minimal mobility distortion and it's symmetric and everything. It's a beautiful task, a beautiful situation. We keep the mean the same, so everything is under control, okay? This is the same Stiglitz that was the chief advisor to the World Bank at one point, and a Nobel laureate and everything. Okay, so our utility function here, we plot our two gambles, the blue gamble and the red gamble, and the x-axis. And then we project that onto the utility function. We draw a line and then go over to the y-axis in order to get the mean between the two blue points called the expected utility and the mean between the two red points, but of course in the metric of utility called the expected utility. And you see that the expected utility for red gamble is higher than for the blue gamble. And as I argued that a mathematical function has predictive value and utility was made to explain choices, this should explain or predict the choice. And here it is. Not fantastic, but it's very clear. The animal prefers the red gamble over the blue gamble, although the physical value, the mean, the expected value is absolutely identical. And as you can figure out what I'm aiming at, the neurons do the same thing. The neurons respond stronger to the riskier gamble than to the blue gamble. This is not a risk response. This is a utility response, or a response in which the physical value is modulated by risk into a common utility signal. And this is what the dopamine neurons are doing. I don't want to say that this is a risk signal. It's a risk modulated value signal, or in conceptual terms, it's a utility signal, right? That we see. Right, maybe that was my slide here. I was looking forward. We're now trying to use the gambles and their neuronal responses to give it into a model, into a TD model, to see if we get CS responses, reward predictive responses, expected utility responses that would be similar to the actual neuronal responses. So here are the gambles. They're now plotted from the top, the blue and the red gamble. And you see on the left-hand side, the same graph here, this utility. We take the neuronal responses from single trials. And you can imagine that the response to the 1.2 milliliter reward is much higher than to the, for example, 0.5 milliliter reward. That's what the graph shows. Then we put that into a model. We randomly vary a little bit, or we take the single trial responses. In fact, we have two distributions, the red distribution and the blue distribution. And sorry, the red and the blue distribution are the CS responses already that we come out of the model. And then we compare that with the graph we just showed. And that again shows that the response to the red gamble is higher than to the blue gamble. So in principle, these responses to utility would be able to produce learning of an expected utility CS stimulus response in dopamine neurons. That's the idea of that graph. So conclusions. I'm not going through the boring details. Artificial dopamine stimulation indicates behavioral neuronal reward function of dopamine utility prediction error signal. This is what we showed with the optogenetics in the monkey. I should say Trisha Janak had very similar results in the rat, and many other people had also similar responses without the utility. in mice and rats with optogenetics. But there are a few things that create headaches to everybody talking about dopamine. The first one is the multi-component nature of phasic dopamine cycle. I mentioned that before. If one is not careful, one has some stimuli, some reward in the laboratory somewhere. Any animal in the laboratory usually expects reward, and that may generate inadvertent responses of dopamine neurons that seem like a totally different attentional response or something. The second thing is this multiple kinds of phasic dopamine activation that people are arguing forever about it, in particular in the last several years. And there were several papers in which optogenetic stimulation would produce different kinds of behavioral activation depending where and how long in the trial they stimulated. And the third thing is what I realized actually, that this TD error coding is not an easy thing to capture. You need to go to extreme lengths to really dissect it, get it out, like Kimura did in what was it, 2011, in this very nice PNS paper, to figure out where is this TD error? Is there a chance that these dopamine responses that are so complicated are actually simply reflecting this TD error? And then, of course, my last slides showed that what I wanted to say, this subjective dopamine coding, this utility prediction error signal, and it is able to update the CS responses for expect utility. And the monkeys are expect utility maximizers, and so are their dopamine signals. We have a few more ideas about that, but I think I should stop here. So the multiple dopamine functions for the single brain system called dopamine, as heterogeneous as it is, admittedly, challenge the conventional concepts of single brain function or single structures. So there's a lot of work to do. So, all right, we have all these crazy ideas here that I went through, and we can dismiss all of them. In the end, find the real function of dopamine. And I'm forever grateful to the organizers to having organized that conference. Thank you so much.
Okay, great. Thanks so much, Wolfram, for a great talk. Lots to think about. So we have a number of questions from the audience, and I'll hand it over to Ben to field those.
Can I end this?
Thank you, Wolfram. First question. This is a question from an anonymous attendee, and the question is about the ability of dopamine neurons to co-release different neurotransmitter. Many of them release not only dopamine, but also glutamate, and maybe also GABA. And the question is, do you speculate about how this co-release could be related to possible different functions of dopamine that you talk about?
Of course. We are doing a very simple initial step. We're looking at the spike responses, right? The spike responses then will release dopamine, and they will release many neurons, other neurotransmitters. And then there will be a presynaptic influence on the dopamine terminals in the striatum or cortex or wherever. That then modulates the dopamine release. So we have a signal that is changing along the way. And then, of course, with the different transmitters, we're talking of different receptors. So then between the dopamine spike and the release through presynaptic interaction, in addition of dopamine and the other transmitters, also, then to the receptors in the postsynaptic neurons, and then the postsynaptic neurons being heterogeneous and having a D1 and D2 pathway and things like this, you get a very complex system, right? So we have to take all of that into account if you want to talk about the dopamine function. It's not an easy thing to do. We are only, from our side, I mean, it's a convenient experiment to record the activity, the electrophysiological activity of dopamine neurons, but I'm very well aware that we're not looking at the whole picture.
Thank you. Switching gears and now talking about learning, there's a question by Genoveva Uzunova, and the question is about the relationship between neuronal learning and behavioral learning. How, for example, how do the changes in the activity of dopamine neuron that is related to learning is related to the behavioral learning itself, and whether if there's any lag between the two, would that be an explanation or sort of an intuition about why there is an extended effect of drugs acting on dopamine receptors?
Well, the behavioral learning would be the net effect of the dopamine action And the net effect of dopamine action relies not only on the US response, but also on the CS response. And that is a neuronally learned response, right? This is the CS is the higher, at least according to the TD model, which works to a good extent on dopamine neurons, is a higher order reward. So the behavioral learning is a result of the, let's say, almost unlearned, although I'm exaggerating. The US response is not an unlearned response either. We can discuss that. And the learned neuronal response. So the behavioral learning is the downstream effect of neuronal response, including neuronal learning.
Thank you. I have another question, which is, you've discussed about, this is a question from Ali Ghazizadeh. He asks, how do you separate novelty from reward uncertainty? And I would like to expand on that. And in general, there are many different components that seems to be in some ways correlated regarding utility, risk, uncertainty, and novelty. And how much do you think it's actually important to try to separate or dissect these different components?
Well, it depends how you want to characterize your signal, but novelty and uncertainty is not the same thing. In particular, in our situation, in which we have 10,000 trials in which there is a gamble with the top and the bottom. There's nothing novel about it. It's simply surprising. You don't know in the next trial whether you get the top or the bottom outcome, right? You could even argue, as Gershman and Uchida had done in a very nice paper, you might develop a prediction of the top outcome and the prediction of the bottom outcome, and that you calculate the prediction error relative to these two predictions. A very interesting idea. But anyway, the attention, of course, attention is generated by novelty, attention is generated by risk, attention is generated by prediction error. So attention is generated by a lot of things. I think I still like to separate these things and try to characterize them as much as possible. I'm not sure I answered the question completely.
No, I think that answers it. Another question from Ryan Huge, and the question is whether did you see any motor effects from your optogenetic activation in the monkeys, and in general, do you see any behavioral effects other than those that you described in your slide?
Behavioral effects of what?
Or for example, motor effects from the optogenetic activation of dopamine neurons in the monkeys, maybe similar to what I've been seeing in mice.
Yes, I'm not entirely sure. We may not have looked at that. We were particularly concerned about the choices and to affect the choices. And it was not always easy to find the effects because the electrical simulation is so much stronger. We used that kind of as benchmark. And then the optogenetic simulation was so much weaker. We were more concerned with the technology of getting the optogenetic simulation working. I do not recall, but it may well be, Bill Stauffer did the experiment, that there is something. So Bill is now at Pittsburgh. He did an unbelievable, great job, and just drop him an email.
Okay, and I would like to just expand on that as a follow-up, personal follow-up. So you've talked about the relationship of dopamine neurons to movements at many different time scales. There is this sort of tonic activity that you can see how basically in Parkinsonian patients, if you give them L-DOPA, they restore some of the movements. So there is this very tonic, very slow effect. Then you have this, what you call the slower effect in order a few minutes. And then people have also described a faster effect, which you call a behavioral activation. So how would you sort of, what would you think about putting all these things together specifically about movements and the different time scales that dopamine seems to affect?
Yeah, I don't know. I mean, what we have observed and never published was that when we did learning studies, the tonic activity of dopamine neurons, just the background activity was higher. It was about 5 to 6 or 7 impulses per second for the whole experiment, as if the monkey was more agitated during these learning situations. That was many years ago. We did a lot of learning situations, not very well controlled. So the animal struggled to learn. It took weeks for the animal to learn the task. We were recording dopamine neurons every day. And we just were surprised at how high the dopamine activity is. So I think the time scale can be anything from less than a second to, well, in this case, several weeks or so, well, at least in the laboratory, let's say a couple hours of elevated activity. And if you look at the old papers in which they had aversive stimulation in rats, they had activity for lasting for 30 minutes or something. Sometimes they were measuring dopamine release or even dialysis, which of course has a time base that's a little slower, like a second, half a second, or to several minutes. Sorry, half a minute to several minutes, and they saw elevations over several minutes, or several tens of minutes with dopamine concentration measured by dialysis. I don't know. I think one could really do nowadays one of these experiments to separate these things. And it, of course, what now Uchida showed is a part of that. But I think we saw more than in his very well controlled and very concise tests. For example, we saw some tonic activity after the reward when the animal was kind of chewing this thing and using his hand and playing around with this piece of apple. At the time, it was food reward, which already has much more stimulation because the animal has to take it also than the liquid reward that just requires licking. So I think there's a lot of going on, and one of my collaborators once suggested to make a comprehensive study of these slow activations.
Thank you. Another question that we have by an anonymous attendee is to try to, do you think there's any differentiation between signals that are behaviors that are learned, rewarding behaviors that are learned, null behaviors that are learned, and innate behaviors? Do you think there is sort of a different utility prediction error for things that the animals basically grows up with, or for things that are learned in a lab context, for example?
Yeah, I think we initially, when we did these very complex tasks, and in fact, the very first task that I presented that was published in 1983 in Experimental Brain Research, we didn't see any reward response. And Malin DeLong at the same time published a paper in Journal of Neuroscience and didn't see any reward response. And he came to me and said, how do you see that later? How do you see that reward response? And I think we just had too many things that were going on and on and on. And there was a continuous slow modulation of reward prediction where there was no really sharp prediction error. So you can easily miss that reward prediction error. And then we thought, well, this is all too complicated. We want a natural thing. And then we started with that food box in which the animal reached. And then we saw much better responses. But that is an almost innate thing. The animal reaches forward to grab a piece of food in front of it. That's not innate, but it's very, very intuitive compared to an arbitrary eye movement, oculomotor choice between two risky stimuli or something. And we didn't see much difference. I think the prediction error is a very solid, basic response that is very difficult to get rid of.
Thank you. I have a question from Priscilla Brossi. This is about one of the slides. If you can go back to the slides, one of the utility slides where you show the higher activity for red activation versus for the red stimulus versus the blue stimulus. And in one of those slides, there seems to be multiple components. No, if you can go back, please. Yes. Please go back again. And I remember seeing that slide as well and thinking about it. There seemed to be sort of a more sustained response to one of the initial slides about utility. I think you should go back more. Seems to be a more sustained response, almost like a multi-component. No, I think we went past it. I'm not. Yeah, no, I think we went past it.
Okay.
Yeah, I should have wrote down the slide number. Let's go forward. If we won't find it, then we'll skip to the next question. But in one of the slides, there seemed to be sort of you had the gamble where there were more there was higher activity.
Yeah, that would be the part of the talk. There would be more.
Yeah, here. There seemed to be.
Not this one, but there was.
No, it was a multi.
Maybe maybe this one. Maybe that one.
That one.
Well, the green one has a yeah, I don't know. Yeah, it's a rebound.
Yeah, never mind. So in general, the question is, how do you think that these signals are recruited?
That's multi-component here, but I have no explanation for that.
Yeah, no, this is it. This is it. Perfect. So you can see in the red that even though the peak is higher on the left side, there seems to be sort of a sustained response beyond that peak. And the question is, what do you think? Are you seeing the left side?
This is maybe 20 neurons on average. I don't know. I don't know.
Yeah, and the question is, do you think this relates to maybe different population of neurons that are recruited later in time?
We would need to see if that happens also in the other monkey, if it's systematic. I don't know. I don't know. There's clearly noise in the system.
Sure. All right, I think we're out of time for questions. There are many questions. We'll post all of them in the Slack. Thank you so much for the great talk.